Today we will be discussing DNA replication enzymes and their use in the biotech industry. And so our learning objectives are as follows: that at the end of this lecture, one should be able to describe the role of each one of the DNA replication enzymes, and I'm not including all of them. We're going to discuss helicase, DNA polymerase, topoisomerase, Taq polymerase, reverse transcriptase, and T4 DNA ligase. These are very pertinent to what we're doing in the lab, and so it's very important that we discuss them. Now, please keep in mind that Dr. Garner is going to expand on this when he talks about cloning. Let's talk about each one of the DNA replication enzymes. Helicase is the one that breaks the hydrogen bonds and separates the two strands of the DNA template during DNA replication. Topoisomerase is the enzyme that goes in front of the replication fork and relaxes the kinks in the DNA so that replication fork can proceed through without any problem. DNA polymerase 3 is the one that comes in after primase lays down the RNA primer and begins to synthesize DNA. The leading strand is synthesized in a continuous manner, lagging strand discontinuous manner. After DNA polymerase 3, then DNA polymerase 1 comes in and extends the existing DNA fragments and then removes the RNA primers. And then DNA ligase comes in and connects the fragments together. That's the last step. Taq polymerase is a DNA polymerase that can act at its optimum temperature is 72 degrees centigrade, and that's why it's very important in DNA amplification. And of course, telomerase, which is a reverse transcriptase, is the enzyme that copies the 3' end of the chromatid or the chromosome to make sure that it doesn't get shortened with each mitotic cell division. So let's begin. Helicase. How is helicase used? Well, I'm just going to describe one use for the helicase enzyme, and that is for the amplification of DNA template, just like we do in the PCR. Now, here this consists of amplification of a DNA template, helicase in the mixture, which will separate the DNA strands, and then the primer, which is also added, is bound to the sense and antisense strand, and then the DNA polymerase, which is also in the mixture, will begin to extend the primer. Okay, so and then after one round of amplification, you have two copies of the DNA that are identical, just like in PCR. Now, the difference between this and PCR, of course, is that you have a helicase enzyme, which is not in the PCR reaction. You have a DNA polymerase, which is not the same as the Taq polymerase. This DNA polymerase actually acts at one temperature and one temperature only, and that is 37 degrees. So, since helicase and DNA polymerases are from either bacteria or humans, they can actually act at 37 degrees. So the difference between this and PCR is that you can do the DNA amplification at one single temperature. You got denaturation, annealing, and synthesis at exactly the same temperature, whereas for the PCR you have the denaturation, annealing, and synthesis, which are at different temperatures. Okay, so this is one use of the helicase, and it is used in helicase-dependent amplification. Then we come to the DNA polymerases. Let me just review for you. There are at least three. There are more than that, but we talk about DNA polymerases 1, 2, and 3 from the prokaryotic system, E. coli, and here is the difference. So I'm just going to jump to the rate of polymerization. All of them have the 5' 3' polymerase activity, and DNA polymerase 1 adds about 60 nucleotides per second, 20 nucleotides per second. The DNA polymerase 2 adds about 10 nucleotides per second, and DNA polymerase 3, which is the DNA synthesis enzyme, adds about 1,000 nucleotides per second. All of them have the 3', 5' exonuclease activity, which is the proofreading activity, and only DNA polymerase 1 has the 5', 3' exonuclease activity, and this comes in handy when it begins to remove the existing RNA primer during DNA replication. 5', 3' exonuclease activity is absent in DNA polymerase 2 and DNA polymerase 3. Now we're going to talk about the use of DNA polymerase 1 and DNA polymerase 1 only in the industry, in the biotech industry. The way that I'm going to describe here, one of the uses for DNA polymerase 1 is in DNA sequencing. Now, let's assume that you're going to sequence a template that has the following sequence, right? So what you do is you mix the following. In 4 tubes, you set up 4 tubes, and in each tube, you're going to mix in DNA template, which is denatured, primer, that is radio-labeled at the 5' end with P32 is often used, and then DNA polymerase 1 is also added, and in each one of the 4 tubes you have deoxynucleotide triphosphate mix, which has DATP, DGTP, DCTP, and DTTP. You add all of these together. Now, each tube is labeled either A, G, T, or C. To the A tube, you add a dideoxy ATP, all right? And they all have this. They don't have the 3' OH. So once this is incorporated, that chain is terminated. It doesn't grow, okay? So the A tube that is labeled A is going to contain a DD ATP, and G, DD-GTP, C will contain DD-CTP, and T tube will contain DD-TTP, all right? And so each one of these is allowed to carry out the reaction. DNA polymerase then extends that primer. The primer is annealed, and then that primer is extended. Okay, and after that reaction, all the reactions are terminated. We're going to look at an example here for A. Now, let's assume, for the sake of simplicity, that you have a primer that is a 1 to 4 nucleotide long. Usually about 18 to 22 nucleotides, but we're going to use one that has 4 nucleotide long. At the 5' end, you have a P32 label right here to make it radioactive. So this is going to bind to the 1, 2, 3, 4 right here. Okay? So I'm going to leave that arrow right there. So let's say that we're just going to look at the A tube. Okay? Wherever there's an A, that's wherever there is an A that's supposed to be synthesized, so you're synthesizing a complement to this DNA strand right here. Wherever A is to be inserted, if that dideoxy ATP is inserted, it's going to terminate that chain. So every conceivable location for an A in the chain is going to be terminated. So let's look at this. You got 1, 2, 3, 4. Now I'm just going to count. 5, 6, 7, 8. The A is inserted here, complementary to T. 9, 10, 11. A is inserted here. 12, 13, 14, 15, 16. A is inserted here. 17 and 18. So the sizes will be 8, 11, 16, and 18. Okay, all of these fragments are synthesized in abundance, of course, inside the A tube, okay? Because the A tube contains not only all 4-deoxy nucleotide triphosphate, but it also contains dideoxy ATP. So that chain is going to be extended, and by chance it's going to be terminated at every spot where an A is supposed to be inserted. So all these can be electrophoresis to separate them on denaturing acrylamide gel. And the reason why denaturing acrylamide gel is used is because acrylamide gel has a differentiating ability to separate two nucleotide chains or fragments that differ by one nucleotide or one base. And so if you have a 16 nucleotide fragment and a 17 nucleotide fragment, those will be able to be separated on a acrylamide gel as opposed to an agarose gel. Agarose gel cannot discriminate between those two. Okay. If we go here, this is really just example where all the 4 tubes are set up. So you have 4 tubes are set up. This is A, T, C, and G. All right. They're all set up, and each tube contains the denatured DNA template that is to be sequenced, and then it's got a 4 nucleotide primer, and it is labeled at the 5' end with P32. In addition to that, you have DNA polymerase 1 that is added, and in addition to that, you have excess amount of all the 4 deoxy nucleotides: DATP, TTP, CTP, and GTP. So all of these mixed together in each one of these tubes, and then each one of these tubes contains the dideoxy A in tube 1, dideoxy TTP in 2, CTP in 3, and dideoxy GTP in tube number 4. And so you let that reaction proceed, and after a few minutes you terminate it. And so let's look at this. Let's look up here. So everywhere that an A is going to be inserted, it's going to be, all the fragments are going to be generated right here. Okay, so let me count again. You have so after this you have 1, 2, and 3. You have 1, 2, 3, 4. Okay, 4 of these, 5, 6, and 7. 6 and 7 are A's. 8, 9, 10, 11, 12, 13, 14, 15. So 6, 7, and 15 fragments are going to be generated. Okay, this is where all the possible A's could be terminated right here. All right, now we go to the next reaction, and that is TTP. So every place that a T should be inserted, that chain is going to be terminated. So we have this is 4, 5, 6, 7, 8. So this is 9, T goes here, 10, T goes here, 11, 12, 13, 14, T goes here. So 9, 10, and 14, and these are the fragments. 9, 10, and 14 fragments are generated in the T reaction. Okay, now let's go to the C reaction. The C reaction, everywhere that a C should be inserted in the complementary strand, the chain is going to be terminated. So you have, let's see, 4, 5, 6, 7, 8. That's a C here. 9, 10, 11, 12. C here. So 8 and 12. Fragments of 8 and 12 nucleotide large or in size are going to be generated in this tube. And finally, we come to the last tube, which is DDGTP tube. Okay, everywhere that a G is to be inserted, you have termination of the chain. So 1, 2, 3, 4, 5 right here. G is inserted. 5, 6, 7, 8, 9, 10, 11. 5, 11. The G is inserted here. 12, 13. 5, 11, and 13 is the size of the G fragment. So 5, 11, and 13. Now, you run the gel, which you have generated 4 of these wells. So A well, you're going to run the A reaction, T well, T reaction, C reaction, G reaction. And they're going to separate out based on the sizes that I put the numbers in, right? So the fastest moving one will be 5. Then comes 6, 7, and then 8, 9, 10, and then 11, 12, 13, 14, and 15. So the movement is going to demonstrate the location in the DNA sequence. So here you can just read right here. All here are G. So you read from the bottom, which is 5' N. G, A, C, T, T, G, C, G, T, A. I hope that makes sense, right? So the sequence will be G is the smallest, then comes 2A, A, then comes C, C, and so it matches because we have G and then 2 A's, A, and then a C, C, right? G, A, C, and then 2 T's, so T, T, right? And then G, C, G, and so 10, 11, G, and then 12, C, and then G, G, okay? G, C, G, and then the final 2 are T and A, and let's see, 14 is T, and then 15 is A. And so when you write the sequence out, right, that's what you're going to write out, right? You're going to write out A, and this is the sequence. Let's see, no, G, A, C, T, T, G, C, G, T, A. That is the sequence that you read, okay? And of course, complementary to that will be the sequence of the template right here. And you can see that A, T, T, G, A. And so if you go up here, let's see, sorry, 5 prime. T, A, C, G, C. Let's see, T, A, C, G, C. And then A, G, T, T, A, G, T, T, C, A, C, G, T. And then you have G, T, T. Yeah. So I hope that makes sense, right? So the complement is the template right here, and the sequence that you read is actually a sequence of the opposite strand right here. And so that's how you sequence and you determine the sequence of DNA. And I'm going to move on, and I hope this makes sense. If not, then tomorrow we can diagram it on a board. These days, though, no one hardly use radioactive sequencing anymore. Everybody's using fluorescent dye. And so everything is basically the same. So you have DNA polymerase in addition to the single strand DNA template. And then you have the deoxy A, G, C, and TTP, right? They're all mixed together, plus the dideoxy A in the A tube, G in the G tube, dideoxy C in the C tube, dideoxy T in the T tube. The difference, though, since the primer is not labeled, the difference is that each one of these has a dye that is attached to it, okay? So that the A lane is going to fluoresce green, G lane is going to fluoresce yellow, D lane is going to fluoresce blue, and T lane is going to fluoresce red. And so you have a machine that goes along as they come off the gel and begins to read. And so here let's see, if we had a primer and we begin to sequence, let's say the primer ends right here, and you're going to begin to sequence at synthesize right here. So you're going to have T, and then T, and then G, T, C, G, A, G, T, C, A, G. And that's basically what this is, right? So the smallest fragment would be the primer plus 1 T. Next one is primer plus 2 T's. Next one with primer plus 2 T's and a G, which is here. Next one is a primer plus 2T plus G plus a T, which is next one. The next one is 2T, primer plus 2T, G, T, and C. And then next one will be, in addition to that, will be an G. And then in addition to that will be an A. So you just go down. And so when you run this down, run it out on a gel, the first one that's going to come off is the T, right? Second one that's going to come off is the T. Next one that's come off is a G. Next one that's going to come off is another T, and then C, and so forth. And that's how the machine kind of reads it based on the fluorescence. And so that's the use of DNA polymerase I in sequencing. And so what the machine does, and when we send our plasmids in to be sequenced, and they begin to sequence, and we tell them what primers to use, and after they sequence it, they're going to send us a printout right here, and they're actually going to read them. So you have T, that is red, C, blue, A, green, and then G, black. So you can just go along T, C, A, G, 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 and so that's how you read it, okay? And you're going from the 5' to the 3'. Remember, the smaller fragments are at the 5', and as you go toward the 3', the fragments get better.